So, we are back with DNA Universe, the 30 layers introductory journey. Basically, we will try to build our concept about DNA and about life and about biology. Hope this course will help you. And let's get started. So, what is life trying to do exactly? Well, the answer to this question is gonna be four things. Okay, these four things will answer your question. Life is basically trying to store the information and then use that information to create more new information and then copy that information and then pass that information to the new generation. That is life fundamentally. Why this matters? Because everything in molecular biology exists for these four jobs. When something seems confusing, we want to ask ourselves, which job does this help with? Now we have question. If a cell stores information but cannot copy it, what's going to happen? Well, the cell would die, of course, because the cell would die when it divides, right? The new cell would have no instructions. Copying is essential for its life to continue. The cell. What is a cell? Cell is a giant factory with specialized departments. Cool analogy, right? Imagine cell as a giant factory. It has these walls, a safe room for blueprints, assembly lines, power plants, and workers. Everything, it's like a big city, honestly. The parts of cells. What are these parts of cell? These are DNA, RNA, ribosomes, proteins, membranes, and nucleus. And the mitochondria. Mitochondria is the powerhouse of the cell. That's a very common phrase. What is DNA? DNA is the master blueprint that decides everything. What is RNA? RNA is the messenger or the photocopy of the DNA itself. What is ribosomes? Ribosomes are the protein factories. And what are proteins then? Proteins are the workers and the machines. Now, what are membranes? Membranes are the wall of these factories. Nucleus. Nucleus is the safe that holds the DNA. And what is mitochondria? Mitochondria is the powerhouse of the cell. I hope you know that. Next question. The big question. The entire field of biology answers one question. How does the factory store the information and use them? We are basically trying to understand the DNA itself. Well, to answer that question, we want to seek other questions. First question is that why doesn't the cell just take the DNA directly to the ribosome? Because ribosome is the protein factory. Why? Because we don't want to lose the DNA. DNA is the only copy of master blueprint, right? If it gets damaged, if the DNA gets damaged, the cell dies. So it should and it must stay protected inside the nucleus, right? The messenger RNA, mRNA, carries the copies instead. Okay, so cool thing happening right there. So we have four things happening right now. We have DNA, we have messenger RNA, that's the copy that carries the copy of the DNA, right? Next question. What is DNA? DNA is just not a chemical, okay? DNA is information. We want to think about it differently. Most people think that DNA is a molecule. Well, wrong way of thinking, I think. DNA is just information, right? The molecule is just the medium, like a paper or a medium or like a book, right? Next thing we want to know is DNA can be a book, it can be a computer file, it can be a recipe, it can be even a software code, logically, if you think so. So, what are you exactly saying? Well, I'm saying is that DNA is a very long instruction manual. The chemical part is just the ink on the paper. The content is what really matters, okay? Then the next question arises. The next question is, if DNA is just information, how could you store in a different molecule? How could you do it? Well, you can do it, right? You can store that information on a piece of paper as well, right? So this is exactly what scientists have already done. Yes, scientists have stored the digital data in the DNA and can read it back. So the information is independent of the molecule, okay? And it's the sequence of the bases that is important. So let's get to the basics of DNA. As you can see, this is my DNA. It looks amazing, okay? We're gonna start to understand the DNA very simply. DNA uses only 4 letters, okay? A, T, G, C. A is adenosine, T is thymine, G is guanine, C is cytosine, okay? Now, you must be thinking, he's saying these random words. What are these things? Well, these are nitrogenous bases, right? These are bases. These are very important. These letters are called nitrogenous bases. That's the formal name, okay? But think of them simply as ATGC or ABCD of biology, okay? Next thing we want to know is that these 4 letters are the fundamental of your life, right? With just these 4, you can write instruction of every living being that is or ever existed. A human has about 3.2 billions of these letters, okay? That's a lot of letters, 3.2 billions. So next question is that how can these just 4 letters encode all the complexity of life? Well, same way as binary does, okay? Just 0 and 1 encodes all the computer software, right? Because essentially all the computer software are just 0 and 1, right? It's not the number of the letter, it's the length and the order of the sequence that matters. So same way ATGC, the length and the patterns of these letters matters, right? So let's try to understand the DNA itself, okay? So DNA is like a letter. I want you to think of DNA as a letter, right? It kind of looks like a ladder. It's just that ladder is twisted now. That's it. So 2 strands connected with base pairs and then twisted, right? Imagine a ladder, 2 long side rails connected with rungs, right? So these lines, as you can see, the green line that is moving like that, this is a side rail, and then we have a blue side rail that is going like that, right? In the middle, we can see 2 rungs, not 2, multiple rungs, connecting these 2 side rails. Now these side rails are sugar phosphate backbones of the DNA, and the rungs are the base pairs, right? Now base pairs can be in the pair of AT and GC. I will answer you why just these 2 pairs exist, okay? Just know that these rungs are called base pairs, and these base pairs are AT and GC, okay? These middle things. For example, as you can see, here's the violet one and a yellow one. So this is one base pair. Another base pair is going to be violet and another yellow. Then we can see another base pair, green and red. So these are base pairs. And the side rails are these sugar phosphate backbone, okay? And basically these things are twisted. That's it. Why they are twisted? They are twisted because these things, these rungs between the side rails are actually negatively charged. And not just these are negatively charged. This shape is amazing for the DNA because in only the twisted shape gives the DNA its stability, okay? If this twisting never happened, the DNA would break. So this is like achieving equilibrium, okay? So let's go to the next question. The base pair. The base pair is these rungs, right? We are talking about these rungs. AT, GC, AT and GC. So note that A only binds to T and G only binds to C. A only binds to T and C only binds to T, right? That is very important. You want to know that. Why? Okay, we'll know why soon, but then now that the letter, and then we're going to twist it, right? That's called a double helix. This twisting of the ladder is called double helix, and it was discovered by Watson and Crick in 53. So it's been like 70, 80 years. So you know that it's not so old thing that we discovered the DNA, but still we are kind of new to DNA, right? So let's go to the next point. The next point that I'm trying to make is that why would the nature twist the ladder instead of keeping it flat? The question. Well, the twisting protects the base inside the helix, right? This base pair, it's very important. The twisting protects the base inside the helix away from the chemical damage. The backbone, the sugar thing, the sugar phosphate, this sugar phosphate side rails, right? The backbone on the outside shields the delicate base pair, right? So this is exactly how nature gave it to us, because this is the best way for DNA to be kept. Okay, so we ask you a question. We ask ourselves a question. Why A only binds to T? Because A always pairs with T and G always pairs with C. Why? Why not A and G binds and why not T and C binds? Because it's a good question to think about. Because of the shape. Simply, A is shaped to fit T like a key fits to a lock, and G is shaped to fit C like a puzzle piece. Other combinations don't physically fit. Simple answer. Then we have these complementary base pairing concept. Now what is this complementary base pairing concept? If you know a one strand, you can automatically guess the other one. How? We know that A binds to T, we know that G binds to C, we know that C binds to G, and we know that T binds to A. If you know this one pair, you can think about the next pair, of course, because A only binds to T and T only binds to A. So you just have to match letters. Simple stuff. A only binds to A only binds to T, and G only binds to C. So remember that. Let's go to the next one. Why is complementary pairing crucial for copying DNA? Because each strand serves as a template for the other. Separate them and each can rebuild its partner perfectly. That's how the DNA replicates, okay? I hope you understand this crucial point. Next go. Hydrogen bonds. What are these hydrogen bonds? Well, hydrogen bonds are just bonds that are holding these base pairs. Strong enough bonds to hold them and weak enough to separate, right? We don't want something very weak that it keeps breaking apart, and we don't want some bond that is so strong that it takes ions, it takes years to separate them, right? We want something sustainable, right? So these hydrogen bonds is the bond that keeps these base pair together, right? So imagine having two friends holding hands. That's a hydrogen bond, not a permanent, easy to break, but a real enough to keep them together. So 2 versus 3 bonds. We know that A and T pair has 2 hydrogen bonds, and G and C pair has 3 hydrogen bonds. So GC pairs are stronger. Why this matters? This matters because DNA must stay together. So hydrogen bonds hold these strands, and we want them to open easily as well. So hydrogen bonds can break easily. Now this hydrogen bond is just perfect enough, and it's strong enough to hold them and weak enough to separate them. This is the physical relevance of hydrogen bonds. Then we have a new question. The new question, if a gene has more GC pairs, would it be harder to separate? The answer is yes, of course. Yes, harder. Each GC pairs has 3 hydrogen bonds versus AT pairs. AT has 2. So more GC, more bonds, more energy needed to break them. High GC DNA has higher melting point. That's why. So what is nucleotide? You told me we have some nucleotide. Well, nucleotide. What is nucleotide? The Lego piece of DNA. It's a piece of DNA and it has 3 parts in one. Okay? A nucleotide, the Lego piece of DNA, and it has 3 parts in it. What are these 3 parts, Dagda? A nucleotide is a single unit, and the DNA is made of nucleotide. Okay? So nucleotide is the building block of DNA. Okay, so each nucleotide has exactly 3 parts. We have a phosphate, we have a sugar, and then we have a base. So this is a nucleotide. When I have multiple nucleotide, I can make DNA. Okay, when I have multiple phosphate sugar base pairs, I can make nucleotide. I hope you get that. So what's the next question? The next question is sugar. Sugar, deoxyribose, a 5-carbon sugar that is central piece that everything else attaches to. That deoxy means it's missing one oxygen. Okay, so deoxyribose, this sugar thing, deoxyribose, has phosphate and base. If this doesn't exist, if sugar doesn't exist, so the phosphate and base, these two will not exist as well. Other question, phosphate. Phosphate act as a connector. Phosphate links sugar together to form backbone chain. They're like links between Lego pieces. Okay. Next question, what is the base? The base is A, T, G, or C, right? This is the information carrying part. Important. The identity letter. What makes one nucleotide different from other? So base can be A, base can be T, base can be G, base can be C, right? Sugar is the holding part. It holds the whole nucleotide together. Phosphate, it's like a connector. It connects the sugar to the it's like a connector. Okay, phosphate links the sugar together to form backbone chain. Okay, it's very important. So let's put it all together. So phosphate, sugar, base, connector, identity. Base is the identity, right? Can be A, can be T, can be G, and can be C. One nucleotide connects millions of DNA. Next thing, what is the next question? The next question is, if two nucleotides have different bases, are they otherwise identical? Yes, because the sugar and the phosphate are same in all nucleotides. That's a good question, right? If you think about it, because kind of sugar and phosphate is same in the world, right? But base pair is the thing that gives itself an identity, okay? So that's important. Let's go to the next part, the DNA backbone. This blue string that is going from here to here, right? And the green backbone, this sugar phosphate, right? We already know about sugar phosphate, right? So the railway tracks with letter hanging inwards. This is amazing how it looks. So DNA is not just letters, right? When we write ATG, CTGTA, that's only the bases, right? But the DNA has structure beyond the letters. That structure is the backbone. That backbone is the framework. Sugar phosphate chain, this chain I'm talking about, okay? So sugar phosphate, and then we have these base pairs, AG, GA, okay? The alternating sugar phosphate chain forms the backbone and base hang inward. So this red thing is the base. This is the information. This is the identity. This blue thing is the sugar phosphate backbone. It is just carrying inward, okay? So two backbones. We have two backbones, as you can see right now. So DNA has two backbones, one on each side, and they run in opposite directions, of course. So blue goes from up to down, as you can see, and the green one actually goes from down to up in an opposite direction. Now this information, this piece of information that DNA has two backbones and one on each side, it's very crucial to understand the concepts later on, right? They run in opposite direction. The base reach inward from both sides and meet in the middle. So I hope you know that. I hope you understand this concept, because this concept is super duper important. So I'm gonna say it again. Sugar phosphate, this backbone is going from up to down. So one backbone is going in this direction from up to down, and another one goes in different direction from down to upward, okay? Different directions, opposite directions. Now let's think about it. Why do bases have inward rather than outward? It's a cool question, it's a simple question. Because we want to protect them. We want to protect these base pairs, these AG, GT, these things, we want to protect them, right? So that's why they're inside. Anti-parallel strands. Two roads, the concept which we talked about, that these backbones are going in different directions, right? Let's talk about them. So anti-parallel strands. Two roads, cars are moving in opposite directions, right? Anti-parallel strands, they scare the students. It's actually super duper simple, right? The DNA strands run in opposite direction, like this, from up to down and from down to up. Simply, this is the concept. Now, let's name them. Simple name them. So one strand runs from direction 5 to 3, and the other strand, this other backbone, runs from 3 to 5, right? The other strands run like this, from down to up, and one strand goes from up to down. Simply, these two lanes of highway, the cars are going opposite direction, okay? So this is simple concept. The critical fact here is that DNA can only be built from direction 5 to 3, from higher value to a lower value. Just remember that. This single fact creates a leading strand, a lagging strand, and Okazaki fragments. We're going to talk about these Okazaki fragments soon. So everything about replication asymmetry comes from this one rule. The one rule is that anti-parallel rule, right? They are not parallel. They are not two roads in same direction. They are two roads but in opposite direction. One road is from 5 to 3, and another goes from 3 to 5, okay? So, next question. Why can't DNA polymerase work in both directions? Because of the chemistry. The enzyme adds nucleotide to a 3 end only, okay? So I want you to imagine I want you to imagine a brick, okay? I want you to imagine a stone, maybe, right? So a Lego brick has an empty end and an end that can be inserted inside, right? So you can only add a Lego brick to an empty end of another Lego brick, right? You cannot add this Lego brick to to that end. I hope you are understanding what I'm trying to get to, right? You cannot join two Lego bricks together without inserting one into another. You have to insert one Lego brick inside another Lego brick, right? So this is the concept that I'm trying to get to. Next thing, why does DNA exist? Basic question. Why it even exist? It exists to store instruction, right? To store instruction and make proteins. Information alone is useless, right? So DNA stores information, but information alone does nothing. This is a recipe book that nobody reads right? A recipe book that nobody reads won't bake any cake. We want the bakers to bake the cakes, right? So a cell needs proteins, the actual bakers, the actual workers, right? Just imagine you just have one DNA. What's gonna happen? Nothing. You need a full heck machinery, right? You need a full machinery. You need people to work on that DNA. You want readers. You want these. You want people who understand DNA and then want to mimic that, right? Just piece of information is not gonna do anything, right? So the cell needs protein. These protein do the actual work, like digest, make hormones, contract muscles, blah blah blah. Make these softwares like this. Everything is done by proteins. Like speaking, everything's done by protein basically. So everything is done by proteins. The connection, DNA stores the information, DNA stores the instructions for making proteins. The DNA is the recipe, proteins are the cooked dish, okay? So think about it. If a gene is damaged, what's gonna happen? We're gonna have bad proteins, right? It depends. A small change might alter the protein slightly. A major change might produce a non-functional protein or a none at all, right? This is the basics of genetic disease. Okay, so basically when we have genetic damage, when we have damage in the DNA at these base pairs, at these A, G, T, when we have damage there, we're gonna have genetic diseases. So gene, what is a gene? Cool question. Gene is a recipe in a cookbook of DNA, okay? Gene is a piece of DNA that codes for a specific protein. That is gene. For an example of a gene is one small DNA segment. So, for example, this pattern called ATG, GAA, GCT, CCA. Now, this is a pattern. This is a specific pattern. Now, this specific pattern is called a gene. And this gene might code to a hemoglobin. Or maybe another pattern called ATBB, not B, ATGA, GT, some pattern might code for insulin. Some other gene might code for collagen. So, I'm trying to learn gene by giving you this example, right? Gene is basically a pattern of these letters, simply. So, cookbook analogy. Gene is the recipe, right? DNA is the cookbook itself. The cell is the kitchen, and the proteins are the dishes, the end product of the genes, okay? So, how many genes do we have? We have a lot of genes. We have 20,000 to 21,000 genes, right? And it's not the number, but how they are actually regulated. It's insane. Trust me, it's insane. Let's talk about DNA. DNA stays inside the nucleus because we don't want to lose the DNA simply, okay? So DNA is precious, the only copy of the master blueprint, right? The cell protects it inside the nucleus, but proteins are built at ribosome outside the nucleus, okay? So this blue sphere is the nucleus, okay? These things are the genes, okay, are the DNA, and these big pills, like these big things, these big structures is ribosome. Note that ribosomes are outside this blue sphere, okay? So we need some messenger that communicates this nucleus to ribosomes, okay? So the logical challenge is that it's like having the only copy of a cookbook locked, right, in a vault, but the kitchen is in a different building. So what's gonna happen? You can't bring the cookbook to the kitchen, of course, because it's locked. It's locked inside a vault, right? Because we are afraid that we might damage the original recipe. We don't want to damage the original recipe. What we can do actually, practically, is take a photo of this original recipe, take that photo, go to the kitchen and cook. That's what we're gonna actually do. The question is, how do the instructions leave the nucleus without moving the DNA, right? The photocopy thing. How do we do it? What would you do if you need a recipe but you couldn't take the book out of the library, click a photo, right? Make a photocopy, the copy of the kitchen. Take the copy to the kitchen, keep the original safe. This is exactly what the cell does. RNA is the photocopy. The RNA is the PDF that you download from Google or from some internet website, right? And that RNA is that copy. The original copy, original book is safe somewhere in some library, locked away. That is DNA, and the copy is the RNA, okay? So RNA, what is RNA? RNA is a photocopy of the master blueprint. RNA is a temporary copy. RNA is a temporary copy. You don't bring the entire cookbook to the kitchen. You just take a copy of some page that you want to, learn, right? So, RNA versus DNA. Let's learn this simple stuff. Our DNA is double-stranded. Double-stranded means these sugar phosphate bonds. RNA is just single sugar phosphate backbone. Okay, base, it has T in it. Okay, base pair. I'm talking about these base pairs inside the sugar phosphate backbone. Right, this. So it has T in it. In RNA, we just have U. Sugar. This is deoxyribose. This is ribose, okay? And lifetime, DNA stays permanent and RNA is temporary because we click a photo and we delete it, right? As soon as we are done with making the protein, we remove that RNA. It's like a useless information now, right? So, why we have U? That T, which is present in the DNA, actually becomes the U in the RNA. RNA uses U, uracil, instead of thymine. So the RNA alphabet is U, AUGC, right? Now a question arises, why? Thymine is more stable, so DNA, permanent, needs stability. RNA is temporary, so RNA just uses U instead of T. It's a simple logic, right? Now, why is RNA single-stranded? Because RNA can be deleted fast, right? We want a temporary photo, right? So RNA needs to be read only by ribosomes. A single strand is accessible and the bases are exposed, so also RNA is super duper temporary, so no need to make it super duper stable. We can just make it simply with a single strand and then just read it fast with the help of ribosome and produce the specific protein, right? So a central dogma, it's a cool thing. A central dogma is a concept, the single most important sentence in the molecular biology. What is it? DNA gives rise to RNA and RNA gives rise to protein. This is how it is. DNA is the main copy, not copy, main thing. Main thing has a copy, that copy is RNA. And then this RNA gives protein, right? But, I think this year, 2026, something has happened in biology where the central dogma has been challenged. I don't know what exactly, so I don't want to mention anything. So step by step, DNA stores the information, RNA copies the information, the photocopy of the DNA, and protein is the final product. So entire biochemistry, everything, replication, transcription, translation, genetic regulation, mutation, blah blah blah, is a consequence of this one flow: DNA, RNA, protein, central dogma. Okay. So can the information go backward? Can we reverse engineer protein to RNA to DNA? What do you think? Normally, no, but retroviruses like HIV can do this. Okay. So retrovirus like HIV can do RNA to DNA using reverse transcriptase. However, proteins to RNA never happens, right? I'm sure it will happen one day and some kind of organism will do it. But what HIV does is that it has this RNA and it creates the DNA using reverse transcriptase. Okay, so that's the concept. Let's go to the next question. What is transcription? Cool word, I think, right? Transcription is just that when DNA is being copied to RNA. Making an RNA copy of a gene, the word meaning writing a copy is like scribe. So copying a document is called transcription. So whenever you see next time word transcription is just DNA is being copied into RNA, that is called transcription. Right? So how it works? It works, as you can see, the DNA, we have this arrangement, right? We have this pattern called ATGCGT. Now you know that we need base pair, right? So how RNA is being copied, right? Transcription is that when we copy the DNA, right? So in RNA, this pattern is going to be written as UACGCA. Note that T in the DNA and U is in the RNA, right? So whenever you see U anywhere, it's RNA. Remember that. Let's go to the next pair. The enzyme RNA polymerase does the job, right? It binds to the DNA at the start of the gene and it unwinds locally and reads one strand, builds complementary RNA, then releases it, okay? Just imagine a machine that maybe binds here, okay? It's localized there. What it does is it stays there, starts reading the base pair and starts going. Read, and then it makes RNA locally, and then when it's done for that specific gene, it actually builds that gene and then throws that RNA, not builds that gene, it builds that RNA and throws that RNA away, right? That machine is called RNA polymerase. So how does RNA polymerase know where a gene is to start, right? That's a cool thing. That thing is called special DNA sequence called promoters. These promoters and terminators, stop sign, mark the gene boundaries. So we have two machines. Two machines, which one? Promoters and terminators. So promoters and terminators, these machines, these DNA sequences, they tell that, hey, RNA polymerase, come to this section, read this specific DNA and make a RNA copy, okay? Translation. So we knew transcription. What is transcription? Transcription simply DNA to RNA copy. That is transcription. What about translation? Translation is that converting that RNA language into a protein language, right? So translation, what is translation? Translation, RNA is transferred to a protein. We make the protein using that RNA, right? So RNA uses 4 letters, right? Protein uses 20 amino acids. So we need to convert that 4 letters of RNA into proteins, right? That process is called translation. So RNA, we have this sequence AUG, GCU, UUA, right? This pattern specifically writes for methionine, alanine, and leucine, right? So AUG writes for methionine. This pattern AUG writes specifically for methionine. GCU writes specifically for alanine, right? So RNA language, we want to translate that into protein. That translation is called translation itself. Okay. So key point is that transcription changes the format, right? Because DNA, we are copying DNA into RNA. So T changes into U, right? But translation changes the language itself, right? Okay, I hope you get that, right? Translation changes the language because we are actually getting methionine, an amino acid, from these simple base pairs, okay? So translation changes the language. Okay, why is it called translation? Because it is literally translating between 2 languages. Nucleic acid are 4 letters, right? And these A, U, G, right? These 4 letters. And amino acids are like 20 amino acids, right? So they are like translating English to Chinese. I hope my analogy is being understandable, right? Okay, ribosomes. What are ribosomes? These are the protein printers. You feed the RNA into ribosomes, okay? So ribosomes read the RNA. It doesn't really read the RNA, it reads the mRNA. What is mRNA? We will understand. So ribosomes make protein. Think of it as a protein printer, okay? So this whole big machine right there is a ribosome, okay? It reads what? It reads mRNA, okay? So input, we put mRNA, okay? The RNA proceeds like read 3 letters at a time, right? It can read 3 letters at a time. We have 4 letters in RNA, of course. So it can do 3 letters at a time. Output is the protein. So we put mRNA here like this, and then it reads 3 letters at a time. And then on one side, it takes the mRNA. On the other side, it produces proteins. two parts. The ribosomes have two subunits, a large subunit and a small subunit. So this is a large subunit, this is a small subunit, right? So large subunit is where the protein is built. This is where the protein is made. Small subunit is where the mRNA is read. Here, okay? They clamp around the mRNA and slide it along. So mRNA actually goes in between here. Think about it. Are ribosomes in the nucleus or outside? What do you think? Of course outside, because we want to keep the DNA safe, right? So outside. So ribosomes are actually outside, right? So outside in the cytoplasm, right? Or on the rough endoplasmic reticulum. It's another address, right? So this is why mRNA must travel from nucleus to the ribosome. Now, what is mRNA? Because I just told you, we'll talk about it. Now we are going to talk about mRNA. Ribosome can only understand mRNA, right? So mRNA, what is mRNA? mRNA is a messenger RNA. Its one job is to carry that information from DNA, right, to the ribosomes, right? So here's the flow. DNA, here's we have a DNA, and then this DNA is converted to RNA, and that RNA is converted to mRNA, and that mRNA goes to ribosome, and then this ribosome makes the specific protein for your requirement. So the journey created in the nucleus. What is transcription? DNA changes to RNA. That is transcription. Exits to the nuclear pore. Now this transcription from DNA to RNA, it's in the nuclear pore. Now this RNA travels to the ribosome. Now this RNA gets translated, mRNA gets translated in the ribosome, and then we get the protein, and then eventually the mRNA gets degraded, right? That's why the RNA has a single strand. Nothing else. mRNA doesn't build anything. It doesn't catalyze anything. It's just a messenger, pure information, okay? Why must mRNA be temporary? Because if mRNA lasted forever, the cell would keep making the same protein when it no longer needs it, right? So degradation allows the control. When mRNA is destroyed, the production stops, simply. So mRNA is just a recipe, right? tRNA, wow, amazing thing. tRNA, what is tRNA? tRNA is just transport, okay? The delivery truck that brings the amino acid, right? So tRNA, transfer RNA, think of it as a delivery truck. What is it delivering? The delivery truck that brings amino acid, important concept. So how it works? So ribosome says, I need methionine. tRNA is like, okay, I have methionine. Then methionine is delivered to the big fat ribosome. Ribosome say, hey, I need alanine. So tRNA say, here, take the alanine. So tRNA is a truck that carries these amino acids for building specific protein, right? So protein grows one amino acid at a time. So key feature, each tRNA has an anticodon, and right, let's understand this. So each tRNA, the delivery truck, has an anticodon. What is an anticodon? An anticodon is a 3-letter code matching the mRNA codon, okay? So that's it actually. An amino acid attachment file, it has this thing as well. What it does, it carries the correct amino acid. So amino acid attachment site is just for carrying a specific right amino acid. An anticodon is to read that specific mRNA requirement, okay? Think about it. How does the right amino acid get attached to the mRNA? It's a cool question, no? It's because of an enzyme called aminosyl tRNA synthase. One of each amino acid, they recognize the specific amino acid and specific tRNA, like quality controls inspector, which is present on the tRNA itself. Okay, so codon. What is codon? These 3 letters, right? What is codon exactly? So codon, these 3 letters, right? One amino acid genetic code, right? Reading in 3s, right? RNA is read 3 letters at a time. Yeah, RNA can be. Each group is of 3, right? So okay, basically what really this thing is saying is that AUG, UAG, right? GCG, these are just RNA, right? So when we read them, this is how the codon looks like. This is codon, METLEUALA, right? This is RNA, this is codon. Okay, so codon, AUG, U, amino acid. Then these gets transcription to methionine, phenylalanine, alanine. Stop. The numbers. We have 64 codons. Wow, that's a lot of. And these 64 codons can translate to 20 amino acid. The code is generate, right? Degenerate. Okay, but never ambiguous. One codon always means one amino acid, right? Okay, know that. That's very important. So the genetic code is nearly universal. Sorry, I was drinking water. Yeah. So the genetic code is nearly universal. Bacteria, plants, fungi, animals, everyone has a genetic code, okay? So powerful evidence for common ancestry. Well, it's cool, pretty cool, right? Let's go to the next thing. So why 3 letter instead of 2? Because 4 raised to 2, that is 16, right? 4 squared is 16. Not enough for 20 amino acids, but 4 cube is 64, enough for 20 amino acids. Nature choose the minimum group size that works, of course. Let's talk about replication itself, right? What is happening in the replication? Why DNA replication, right? Before the cell divides, it must copy its entire genome, okay? So that division problem. What's the division problem here? When a cell divides, you get cell A and cell B. Each needs a complete set of instruction, of course. You just can't split as well, right? Because you can't split these pairs, right? So you can't split the ATCG in half. It would lose the information. Half a recipe is useless. So both cells need full set, right? We need to copy both pairs. Okay, the solution. So before division, the cell makes entire copy of DNA. And this is called DNA replication, okay? Human DNA, around 3.2 billion base pairs, replicated in around 8 hours. So it takes 8 hours to copy this amount of information. And it is done with few errors. That is insane. Think about it. How can the cell copy 3.2 billion base pairs without mistakes? It makes around 1 error per 100,000 bases, right? That's very amazing. But proofreading enzyme fix these errors, bringing the final rate to 1 error per billion. Multiple layers of quality control. That is insane. That is amazing if you think about it. We copy this much amount of data and we just make 1 error per billion. That is amazing, no? I think that's insane. So we are now in a DNA replication. That's amazing. We are going to talk about an enzyme. Its enzyme is called helicase. Now this helicase is actually responsible for opening up these DNA, these pairs, right? So zipper that opens the DNA, this is a helicase, this red thing, all right? This is one sugar phosphate backbone. This is another sugar phosphate backbone. This is a leading strand. This is a lagging strand, right? DNA is like a closed zipper. ATGC, ATGC, right? Just look at these look at this sugar phosphate strand, right? In the middle we have these base pairs, right? Okay, let's see. Helicase. Helicase unzips this DNA, right? Helicase breaks the hydrogen bond between these base pairs and separates the two strands. It's the DNA unwinding enzyme, right? Simply, helicase creates a replication fork, a Y-shaped region where replication happens. It travels along DNA, unzipping as it goes, right? So as you can see, we have a Y shape. Say this is like a Y. This is upper part of the Y. This straight line is the lower part of the Y. What prevents DNA from re-annealing after helicase, right? This open DNA can go back together, right? What's happening here? We have something called as SSB, single strand binding protein. They attach to the separate strand and keep them apart, like a bookmark. DNA polymerase, another enzyme. What is this DNA polymerase? DNA polymerase is the star of DNA replication, building the new strand. Once the strand is separated, we need a new complementary strand for each DNA. So for this strand, we need a machine that copies this strand. For this strand, we need another machine that copies this strand. Okay, we need two machines. So on leading strand, we need a machine. That machine is called DNA polymerase, simply. Okay, so one strand is separated, we need a new complementary strand for each. DNA polymerase is the builder. Okay, how it works? Well, A here, it adds to a C because we know that A only binds to T. G here, it adds to C because we know that G only binds to C. It's not that we know, this machine, DNA polymerase, knows that. It builds a complementary strand to each matching base by pairing rule. This is amazing, no? Let's go. The star, DNA polymerase reads template, it means copies, and selects the correct nucleotide, forms the bond, proofreads it, it means checks if it's good or not, if it's correct or not, its own work, and then move forward. All of it is done 1000 base pair in 1 second. How fast this is? This is so fast, no? Right? Now, how does the DNA polymerase proofread? Right? It has 3 to 4. It proofreads in which direction? It proofreads in a retrospective way. It proofreads in a backward direction. Okay, it has 3 to 5 exonuclease activity. It means that it proofreads from 3 to 5. We know that synthesis goes from 5 to 3, from higher to lower. It proofreads in opposite direction. If it detects some mispaired base, it backtracks. It goes back and removes that wrong nucleotide, right? And it replaces that wrong nucleotide. It's like an eraser built into a pencil, right? Leading strand. What is a leading strand? A leading strand is a smooth highway, continuous synthesis, right? This is a leading strand because this DNA polymerase goes from like this, leading strand, right? It goes faster, leading strand. DNA polymerase can only go from 5 to 3, always. And this creates asymmetry. One side is easy, of course. The leading strand is easy because polymerase is going from 5 to 3, right? Like this. But this lagging strand actually goes from 3 to 5. It's opposite. Because I told you already that one strand is going from up to down and then another strand is going from down to up. That's why one strand is leading and one strand is lagging always. On the leading strand, we have this DNA polymerase. So on one strand, the fork moves in same direction as the polymerase needs to go. Result continuous synthesis, of course. But, so that's leading strand, okay? So this is a leading strand. Think smooth highway, one polymerase moving continuously, never stopping. Think about it. Why is the leading strand called leading? Because it leads the way. Because we are moving in the same direction as the DNA polymerase. That's why it's leading. It leads the way, synthesized continuously in the direction of the fork is moving, right? So polymerase move in the right behind the helicase. So this helicase is actually going down, okay? I want you to imagine this. This helicase is going down, and as soon as it's going down, it is opening these two strands simultaneously. And on one leading strand, we have this machine called DNA polymerase. On another lagging strand, we have another machine. I'm going to tell which machine. And then here is different kind of mechanism happening, okay? So helicase is moving from up to down. Can you imagine that? It's going from up to down, and as long as moving, it's opening these strands, right? So lagging strand, the broken road, Okazaki fragments, right? What is it? Lagging, right? On the other hand, on the other strand, the fork moves opposite to the where polymerase needs to go. It can't work continuously, right? Because we know that in one direction the strand is going from up to down, and another strand it moves from down to up. So it creates this leading and lagging strand. So what happens on the lagging strand? The lagging strand actually, we build, we stop, we jump, and then we build. So polymerase must build a short piece of information, then stops, and then jump back, and then build another information, and then repeat. These small pieces are called Okazaki fragment. And this is like 100 to 200 base pairs each, right? So what's really happening is that DNA polymerase is moving from up to down like this, right, with the helicase. But here DNA polymerase is not moving like this, because this strand goes like, this strand is going in an opposite direction as leading, right? So what it does is that it builds, then it stops, and then it jumps to another pair, and then it does another, and then jumps again, right? This jumping and this building, it creates these little fragments called Okazaki fragments. So lagging strand, this is the lagging strand. Think it of as a broken road, repaired piece by piece. Fragments are later joined together. Think of it. Isn't the lagging strand wasteful? No, it's not. It's not a problem because it is unavoidable consequence because of the 5 to 3 rule, right? Cell has evolved efficiently machinery to handle it, right? Because one phosphate sugar backbone is going in this direction and another backbone is going in this direction. Always like that. Primase. What is primase? Primase is the starter motor. Polymerase can't start from nothing, right? Polymerase has to have a base, has to have one side of the base pair, right? Right, it cannot just build information from nothing. It needs to have something to copy it from, right? So DNA polymerase cannot start a new strand from scratch. It needs ADD to an existing strand, right? That primase, that thing is what primase is, right? Like a car without a starter motor. The engine works fine, but you can't start itself. It needs a push. So primase provides push. Primates create a tiny RNA primer, around 10 nucleotide. The DNA polymerase, right? Oh, great, now I can work on it. It extends from primer. It starts from primer. So primer is that starting line where the polymerase starts, right? So lagging strand needs many, right? The lagging strand needs new primer every Okazaki fragment because it's jumping, it's making, it's jumping, it's making, right? So I hope you understand this concept. Why only primase starts from the scratch but polymerase cannot? Because primase can initiate synthesis de novo. De novo means new, okay? So primase can initiate synthesis de novo but less accurate and slower. Division of labor, primase starts, polymerase extends. Simple stuff. Ligase. What is ligase? Ligase is just a glue that attaches these consecutive Okazaki fragments, basically, okay? Ligase glues them. DNA ligase seals the gaps between Okazaki fragments, creating one continuous strand. Think of it as a DNA superglue. The process, RNA primer removed, replaced with DNA. Ligase forms the final phosphodiesterase bond. This phosphodiesterase is the glue that joins these two Okazaki fragments, right? So lagging strand is now one continuous strand. Think of it. What would happen without ligase? We're gonna have multiple Okazaki fragments, and then we will not have the DNA. Lagging strand would remain fragmented. Chromosome would break easily. DNA would be unstable, and the cell would likely die. Mutation. When the code changes for the better or for the worse or for the nothing, right? What is mutation especially? Well, what if ATGCGT becomes ATTGCT? What if the pattern changes? That one letter that is changed here actually is called mutation. So a mutation is change in protein, alters function, destroys a protein, right? Improves a protein, rare but possible, and can be a silent mutation as well. So evolution exists because mutation exists, okay? Without mutation, no variance, no natural selection, no adaptation, nothing. So mutation is the raw material of evolution. Mutation is the reason evolution exists. So, are most mutation harmful? I don't think so. Most of the mutation are neutral. Of course, some are harmful, some are not, some are good, right? What about chromosome? What is chromosome? Chromosome is 2 meters of DNA packed into a 6 micrometer. Okay, that's like packing 2 meters. That's like packing 60 kilometers of thread into a marble. That's a huge packing thing that's going on, right? What happened? DNA is wrapped around a protein. It's called histone. Okay, I want you to imagine that a thread is being wrapped around a stone. That stone is called histone, like thread around a spool or a stone, right? Now each stone wraps around 146 base pairs, right? That's the amount of base pairs each histone has. So that's how it happens. DNA wraps around histone, right? Now this histone is the stone, right? That beads on string. a lot of time people gonna ask you, what is this bead on string appearance? That's just histone, right? Now that histone that is, present with the DNA wrapped around it, it folds further. Now that folding later becomes chromatin, that is 30 nanometer fiber. Later more folding happens, and then we call it chromatin loops, and then even more folding happens, and then eventually we call it chromosome. So that is like super duper condensed. So eventually DNA is inside the chromosome, but DNA is wrapped around histones. Histone is wrapped around chromatin. Chromatin is wrapped around chromatin loops, and chromatin loops are wrapped into chromosomes. That's how it is. Chromosome is just compressed DNA. Chromosome is highly compressed DNA. Humans have around 46 chromosomes, 23 pairs per cells, okay? So every cell has 46 chromosomes. So why not keep DNA as loose noodle? Because we don't have the space. 2 meter won't fit in a 6 micron cell, right? Second, organization. Chromosomes keep related gene together. Third is protection. Fourth is division. You can separate 46 chromosomes, but not 2 meter of spaghetti. Of course, it's more. It's just it's more organized, right? The entire story, the last big story. DNA stores the information. Replication copies the DNA, right? Transcription changes DNA into RNA. Note that the format is same. Here, DNA and RNA both share the common language. That's why we call it transcription, not translation. If I say translation, I am changing the language itself. So mRNA is the carrier messenger of RNA, okay? So ribosome reads that mRNA and produces the protein, right? tRNA, what is tRNA? tRNA brings the amino acid to the ribosome, right? To the fat ribosome. Ribosome say, hey, I need guanine. And then tRNA is like, okay, I have guanine, I'm gonna give it to you. And then ribosome does its thing. What is translation? Translation is RNA converted to protein. Now here the languages are different. It's like changing English into Chinese. Protein is the final product that I'm getting from the DNA, and then protein give rise to life itself. So that is the whole concept of DNA. That's it. You don't need to know anything more to understand whole DNA biology. That's it. So you can take a screenshot of this or something, I don't know. Everything follows. Once these concepts are solid, everything becomes a consequence rather than memory, right? Histones, telomerase, nothing. You don't need to memorize anything, just is simple, right? So you have completed your course. So I hope, have a nice day. I hope you enjoyed this whole course. If you didn't understand something, you can always try to understand it. It's simple.